Hello everyone, and now we are going to see what is MCV in a CBC report. MCV is nothing but mean corpuscular volume. So when you get your report, you see the report and you see that the MCV is being reported as femtoliters in a CBC report. Now how this MCV is derived and why it is very important. So there are different types of channels within the blood cell counter, the red cell channel and the WBC channel. So inside the red cell channel, so assume this is a red cell. Whenever a red cell passes through the electrical poles, whenever the red cell passes here, electrical pulse is generated. So this is the basically Coulter principle. So whenever a red cell is passing through, you get an electrical pulse which is generated. Now the height of this pulse is corresponding to the cell volume. The height of this pulse is corresponding to the cell volume, and this is measured from individual cell. So as several red cells are passing through the blood cell counter, the individual pulses are recorded. So these are millions of red cells passing through the aperture, and the height of each of this pulse is measured now. So at the end you see that the Gaussian curve is plotted and a RBC histogram is derived. The center of this, the average of all these pulses is nothing but the mean corpuscular volume. The average of all cell volume of all the red cells is the mean corpuscular volume. So in this case, the mean corpuscular volume is 75. Mean corpuscular volume, the normal MCV is nothing but 80 to 96 femtoliters. So whenever it is less than 80, we call them as microcytic red cells. So if this is the normal size of the red cell, less than 80 is microcytic, more than 96 is macrocytic. Now this parameter can help us identify the cause of anemia, which is clinically significant. So in the next video, we'll come across MCV with how can we use this MCV with other red cell indices to decipher the cause of anemia.